protein microarrays, and like the DNA microarrays, it can be called as the protein chips or protein microarrays. If you remember, in our previous discussion, we studied that after the synthesis of messenger RNA or for the detection of DNA, the target molecules, they can be immobilized on the solid surfaces or small chips, and then some known molecules, they can be used for hybridization to evaluate that whether our target DNA is present or not. So on the same pattern, now we can synthesize protein chips. So in other words, we can say that they are miniature devices like the DNA microarrays that contain immobilized protein molecules or any other molecules to detect other protein molecules. So इसमें हम क्या करते हैं कि प्रोटीन्स के मॉलिक्यूल को हम इमोबिलाइज करवाते हैं सॉलिड सरफेसेस पे, और फिर इन प्रोटीन्स को डिटेक्ट कर लिया जाता है बाय यूजिंग सम target compounds that may be antibodies. We are going to briefly study it later on. But as compared to DNA microarrays, protein chips, they have certain limitations. For example, in case of DNA, the process, it is well developed, relatively simple, because now we can synthesize RNA or DNA copies by using PCR technique. But this is not possible for protein because proteins, they cannot be synthesized like the DNA. So this is one of the limitation. Another point is that as far as nucleic acid is concerned, it is mainly made up of 4 nucleotide bases. So with reference to their chemical properties, it is not so much diverse or variable. So same reaction can be performed under same conditions, but this is not the situation for proteins because they may be made up of 20 or 22 amino acids. So proteins, they are more diverse as far as their physical or chemical properties are concerned, especially from chemical point of view, they are more diverse. तो एक ही set of condition हम use नहीं कर सकते प्रोटीन की detection के लिए. So these are a few limitations, but still protein microarrays or protein chips, they have been manufactured. As I told you that they are miniature devices that contain immobilized proteins, and how those immobilized proteins, they can be detected. And here different approaches can be used. Like one can synthesize a set of antibodies that are highly specific to the immobilized proteins. So if reaction is there, we can say that a particular protein, it is detected. We have explained number of times that antibodies, they are highly specific in their reaction. So when a particular target antigen or protein is available, it can react with it. And then this reaction, it can be detected by applying different techniques. And before decades, ELISA has been used for the detection of proteins by using specific antibodies. In addition to that, even antigens, they can be used for the detection of proteins in a solution. So, like the DNA microarrays, protein chips, they are also a valuable tool for the detection of wide variety of proteins. And we are now limiting our discussion to this point, that how proteins, they can be purified or analyzed by using mass spectrometry or by using protein chips, although further study can be carried out with reference to their 3-dimensional structure or protein to protein interaction.